Welcome to the Deep Dives. We're the show that journeys into some of the most complex, fascinating subjects out there. We don't just want to understand them, we want to really experience the wonder of discovery. And today, we're tackling one of the biggest questions, maybe the biggest question. How did life actually begin? I mean, it's a question that's stirred up philosophers, theologians, scientists for, well, forever, really. It touches our whole sense of place in the universe. For centuries, the idea was, a spontaneous generation. Aristotle was a big proponent of the thought that complex things like maggots or even mice could just sort of poof into existence from non-living stuff.
Almost magical thinking, really.
Exactly, but that got completely debunked, didn't it? First by Francesco Redi with those clever experiments on meat in jars way back in the 17th century.
Right, showing the maggots came from flies, not the meat itself.
And then Louis Pasteur in the 19th century really nailed it shut with his swan neck flask experiments.
Yeah.
That gave us this fundamental principle, omne vivum ex vivo.
All life comes from pre-existing life, a cornerstone of biology.
But, okay, let's unpack this. That leaves a huge logical gap, doesn't it? If all life comes from life, where did the very, very first life come from?
That's the enigma. That's the core of it.
And that's where science steps in with this framework called abiogenesis. Now, this isn't going back to spontaneous generation, not at all.
No, it's fundamentally different. It's about process.
Right. It's a scientific quest. How did non-living matter, step by step, through increasing chemical complexity, driven just by natural laws, transform into something living? It's about gradual emergence, not sudden creation.
Exactly. Think chemistry, physics, geology, all working together over immense timescales.
So our mission in this deep dive is to navigate through the hypotheses, the experiments that have really shaken things up, and the mysteries that still remain. How did inanimate stuff become organisms here on Earth? Or it maybe even somewhere else first?
It's not just some dusty academic question, is it?
No way. This is about our ultimate origins. It's about the potential for life elsewhere. It might even tell us what alive truly means.
And to really get a handle on it, you have to see how incredibly interdisciplinary it is. This isn't just biology or just chemistry. It's geology, astronomy. They all bring crucial pieces to this giant jigsaw puzzle. And before we even start charting the path, there's that fundamental question, what is life? It sounds simple, but it's surprisingly tough to pin down.
Really? How many definitions are there?
Well, one count found over 123 distinct definitions proposed by philosophers and scientists.
Wow. Over 100 ways to define something we feel like we just know when we see it.
Exactly. NASA, thinking about finding life elsewhere, uses a practical checklist, 7 key traits: cellular organization, reproduction, metabolism, homeostasis, response, evolution. Things like that.
That makes sense for looking for it.
It does. But maybe a more elegant definition, especially for origin of life studies, is this: a self-sustained chemical system capable of undergoing Darwinian evolution.
Okay, break that down. Self-sustained.
Meaning it maintains itself, uses energy, keeps itself going.
And Darwinian evolution.
It reproduces, it inherits traits, and it can change and adapt over generations. That captures the core dynamic, doesn't it? Self-maintenance and self-perpetuation with improvement.
It does. That feels pretty fundamental.
But what's fascinating here is that researchers don't actually wait for the perfect definition before they start working. They tackle specific, solvable problems. How do amino acids form? How do lipids make vesicles? How do monomers link up? The research moves forward piece by piece.
So the definition itself becomes clearer as you rebuild the pathway.
Precisely. The definition of life isn't some fixed starting point we need to agree on perfectly first. It's almost an emergent property of the research itself. As we figure out how to build a system that meets more and more criteria metabolism than replication, than compartmentalization, our understanding of what constitutes life gets sharper.
That's a neat way to think about it. So today we'll hit the early Earth conditions, the building blocks, how information and function arose, getting cells, and maybe even other places it could have started.
That's the plan. We'll cover the key stages and the big ideas.
Okay, let's set the stage then. I want you to really picture this. Go back in time, way back. The Hadean Eon. That's what, 4.5 to 4 billion years ago?
Roughly, yes. The very earliest chapter of Earth's history.
Forget the blue marble image. This Earth was violent, dynamic, maybe even hellish, hence the name Hadean. Think massive volcanism everywhere, constantly spewing gases.
Water vapor, CO2, nitrogen, building that very first atmosphere, and creating intense geothermal heat gradients. A real chemical furnace.
And it wasn't just internal turmoil. The early solar system was messy. A cosmic shooting gallery, right? This heavy bombardment phase.
For hundreds of millions of years, yeah. Asteroids, comets, constantly smashing into the young planet.
Which sounds terrible for life starting, like hitting a reset button constantly.
It could definitely be sterilizing, vaporizing oceans, wiping the slate clean, a major hazard.
But there's a twist, isn't there? These impacts, they weren't just destructive.
That's the double-edged sword. They could also deliver crucial ingredients. Water ice locked up in comets, for instance, and complex organic molecules formed in space, riding in on asteroids and dust.
So paradoxically, the bombardment could both destroy and deliver the goods needed for life.
Precisely. It's a complex interplay, destruction and delivery happening side by side.
And the key window for life to actually get started was probably during a quieter period.
Exactly. There seems to have been a relative lull in the most intense bombardment, maybe around 4.4 to 4.0 billion years ago. Long enough, crucially, for stable liquid water oceans to form and persist.
And we think oceans formed really early.
The geological evidence points to maybe as early as 4.3 billion years ago. Liquid water, the essential solvent for life as we know it, setting the stage incredibly early.
Wow. Okay, so water's there, but something else was missing, or rather present in a big way compared to now. Radiation.
Absolutely. No free oxygen meant no ozone layer shielding the surface. The young sun was blasting the planet with intense ultraviolet radiation.
Which sounds bad for life again.
For modern life, yes, it's highly destructive, but for prebiotic chemistry, that intense UV light was a powerful, readily available energy source. It could drive chemical reactions that might otherwise have been very slow or required other catalysts. Energy was abundant, just in a different form.
Okay, so a violent, watery world bombarded from space, bathed in UV. What about the air itself? You mentioned the gases from volcanoes, but what was the atmosphere actually like?
Now that is a huge point of debate, and it really matters for abiogenesis models. The classical picture, the one that informed the early experiments, was of a strongly reducing atmosphere.
Reducing meaning?
Rich in electron-donating molecules, lots of methane, CH4, ammonia, NH3, water vapor, H2O, and hydrogen gas, H2. Think chemically reactive, ready to build complex molecules.
Okay, like a primordial chemical soup ready to cook.
Sort of, yes. That kind of atmosphere makes synthesizing organic molecules relatively easy, chemically speaking.
But that's the classical view. What's the current thinking?
Well, modern geochemistry, looking at volcanic outgassing models and the likely state of the Earth's mantle back then, suggests something different. Probably a less reducing, maybe even neutral atmosphere.
Dominated by what then?
More likely carbon dioxide, CO2, and nitrogen, N2, with much smaller amounts of hydrogen, maybe some methane or carbon monoxide, but not the huge amounts previously thought.
And what does that mean for making life's building blocks?
It makes it harder, generally. You can still synthesize things like amino acids in a neutral atmosphere, especially if you have some hydrogen around, maybe near volcanoes, but the yields are often lower, and the specific mix of products might be different.
So this isn't just a minor detail. This atmospheric debate fundamentally changes things.
It's a massive shift. Think about it. The early iconic experiments, like Miller-Urey, were based on that strongly reducing model. If the atmosphere wasn't like that globally, then the idea of a planet-wide surface soup forming easily becomes less convincing.
Right? The ingredients wouldn't just appear everywhere in large amounts.
Exactly. This atmospheric problem really forced a rethink. It drove researchers to look seriously at localized environments where conditions might have been different. Maybe reducing conditions existed inside volcanic plumes or around hydrothermal vents on the seafloor.
Places where the right chemistry could happen even if the whole atmosphere wasn't cooperating?
Precisely. And it also boosted the idea of extraterrestrial delivery. If Earth wasn't making enough building blocks itself, maybe they arrived from space in greater quantities than we first thought. It opened up new avenues of research.
Okay, so the stage is set. A dynamic, challenging early Earth. Now let's talk ingredients. Before Miller and Urey did their famous experiment, there was a theoretical idea floating around, right? The Oparin-Haldane hypothesis.
Yes, back in the 1920s. Alexander Oparin in Russia and J.B.S. Haldane in Britain, independently, came up with this foundational concept. It was huge because it moved abiogenesis from just philosophical musing into the realm of testable science, a theory of gradual chemical evolution.
What were the key points they proposed?
Well, first, they assume that reducing atmosphere we just talked about, methane, ammonia, water, and crucially no oxygen, oxygen would just destroy fragile organic molecules.
Okay, an oxygen-free, reactive atmosphere.
Second, they figured energy sources like UV light and lightning would zap these simple gases, forcing them to combine into more complex organic monomers, amino acids, the bits of proteins, maybe nucleotide bases, the bits of RNA and DNA.
The basic building blocks.
Exactly. Third, they proposed these monomers wouldn't just break down again immediately. They'd wash into the oceans and accumulate over vast stretches of time, millions of years perhaps. Haldane famously called this a hot, dilute soup.
The primordial soup. I've definitely heard that term.
It's a classic. And fourth, within this soup, they envisioned further reactions. Monomers linking up to form polymers, proteins, and nucleic acids. Operin even talked about coacervates, little droplets that could form spontaneously, enclosing other molecules like tiny protocells.
Like little bubbles of chemistry.
Sort of, yeah. And finally, they figured the first life wouldn't be making its own food. It would be heterotrophic, just consuming the ready-made organic stuff from the soup. A simpler start.
That sounds remarkably logical step by step, but you said it was theoretical for decades.
It was a brilliant idea, but just an idea, until 1953. That's when Stanley Miller, a young graduate student working with Miller-Urey at the University of Chicago, decided to actually test it.
The Miller-Urey experiment. This is the big one, right?
Absolutely iconic. Miller built this closed glass apparatus, a miniature version of the early Earth's water cycle. He had a flask of boiling water as the ocean, creating steam.
Okay.
The steam mixed with gases in a larger flask, methane, ammonia, hydrogen, mimicking that presumed early atmosphere. And in that flask, he had electrodes sparking, simulating lightning.
Zap.
Constantly zapping. Then a condenser cooled everything down, making it rain back into a collection trap at the bottom. The whole cycle repeated.
Then what happened?
It ran for just a week. And the clear water in the trap turned this murky reddish-brown color.
Something was definitely forming.
Oh, yeah. Miller analyzed the water using paper chromatography, which was state-of-the-art then, and he found amino acids, several of them, like glycine and alanine, the actual building blocks of proteins made from just simple gases and sparks.
Wow, so they actually made the stuff of life from non-living chemicals in the lab.
They did. It was monumental. It showed that forming these crucial organic molecules wasn't some impossible miracle. It could happen naturally under plausible early Earth conditions. It basically launched the whole field of prebiotic chemistry as an experimental science. The soup wasn't just a guess anymore, it was chemically feasible.
That must have been incredibly exciting.
Absolutely transformative. Now, connecting this back, we know now the atmosphere might not have been exactly the mix Miller used, right? More neutral, perhaps.
Right, so does that invalidate the experiment?
Not entirely. Later experiments using different gas mixtures, including more neutral ones like CO2, nitrogen, maybe some hydrogen like you might find near volcanoes, they also produced amino acids. Maybe not always in the same high yields, but they still form.
So the basic principle holds, even if the exact location or conditions varied. Maybe it happened in specific spots, not everywhere.
Exactly. Maybe localized reducing environments were key. And remember the other source we mentioned?
Ah, stuff from space. Meteorites.
Right. Discoveries like the Murchison meteorite, which fell in Australia in 1969, were packed with organic molecules. Over 70 different amino acids, nucleobases, sugars, stuff formed out there in the asteroid belt, or even earlier.
So Earth was getting deliveries too, topping up the soup.
Precisely. It paints a richer picture. Synthesis happening here, plus materials arriving from elsewhere, all contributing to the prebiotic inventory.
Okay, so we've got the monomers, the building blocks from one source or another. But life isn't just monomers floating around. They need to link up into long chains, polymers, proteins, RNA, DNA. And you mentioned this causes a problem, the water paradox.
Yes, this is a really fundamental challenge. Think about how these polymers form. To link two monomers together, you typically have to remove a molecule of water. It's called a dehydration reaction.
Makes sense. Dehydrate to link.
But now imagine trying to do that in the primordial ocean, which is, well, full of water. Thermodynamically, the equilibrium strongly favors the reverse reaction hydrolysis. Water wants to break the chains apart, not let them form.
So the soup itself is actively working against building the necessary long molecules. That seems like a major hurdle.
It is. It strongly suggests that just having a dilute soup wasn't enough. Polymerization needed something more. It likely required specific kinds of environments, structured places where you could overcome water's tendency to break things down.
Okay, so how do you beat the paradox? What kind of places or processes could work?
Scientists have come up with several clever and experimentally supported solutions. One idea involves evaporation cycles. Think about shallow pools near volcanoes or tidal areas on coastlines.
Places that get wet and then dry out repeatedly.
Exactly. When the water evaporates, the molecules dissolved in it get left behind, becoming highly concentrated. This crowding forces them together and makes those dehydration reactions much more likely. Then maybe the tide comes back in, washes the newly formed short polymers around, they stick to something else, dry out again, grow longer.
Like a natural cycle of concentration and reaction.
Precisely. Sidney Fox did experiments back in the 50s showing you could heat dry amino acids and they'd link up into proteinoids, protein-like chains. So wet-dry cycles are a strong candidate.
What else?
Another really powerful idea is mineral surface catalysis, especially clays, like momerillenite. These clays have microscopic layered structures with huge surface areas.
Like tiny flat sheets stacked together.
Yeah. And these surfaces can attract and bind organic monomers from the water, sticking them down. This does two crucial things. First, it concentrates them, pulling them out of dilute solution. Second, the mineral surface itself can act as a catalyst, lowering the energy needed for the polymerization reaction to happen and maybe even aligning the monomers correctly.
So the clay acts like a template or workbench for building polymers.
That's a great analogy. It holds the pieces in place and helps them link up. Experiments have shown that clays like montmorillonite are really effective at promoting the polymerization of both amino acids and, importantly, RNA nucleotides. They can even help form lipid vesicles.
That sounds elegant. It solves the water problem by shifting the action onto a solid surface.
Exactly. It highlights how the geology of early Earth, the minerals available, could have been just as crucial as the chemistry of the water or atmosphere. Structure matters.
Okay, so we've potentially got building blocks and ways to link them into polymers. But this leads us to maybe the most famous puzzle box in the origin of life, the chicken and egg paradox.
Ah, yes. The information versus function problem.
Right. In modern life, DNA holds the blueprint, the genetic information. But DNA can't do much on its own. It needs protein enzymes to replicate it, to read it, to build things.
And those protein enzymes, their instructions, their blueprints are stored
in the DNA.
Yeah.
You can't have one without the other. DNA needs proteins to work. Proteins need DNA for instructions. How could a system like that possibly get started? It seems like an impossible knot.
It does seem like an impasse, doesn't it? And this is where the RNA World hypothesis comes in as a really compelling potential solution. It was suggested back in the 60s by Carl Woese, Francis Crick, and Leslie Orgel.
RNA, not DNA. How does that help?
Because RNA is special. It has this amazing dual functionality that DNA and proteins mostly lack. It can bridge the gap.
Okay, what's the dual function?
First, like DNA, RNA can store genetic information. It uses a sequence of bases A, U, G, C, and through base pairing, it can act as a template to replicate itself so it can handle the information storage job. Check.
All right, information, check. What's the other job?
Catalysis. Doing things. Unlike DNA, which is usually a rigid double helix, single-stranded RNA can fold up into complex, specific 3D shapes, just like proteins do.
Ah, so structure dictates function.
Exactly, and these folded RNA shapes can form active sites that catalyze chemical reactions. We call these RNA enzymes ribozymes.
Ribozymes, RNA enzymes.
Yep. So in an RNA world, you could have RNA molecules that were both the genetic material and the enzymes needed to copy themselves and maybe perform other simple metabolic tasks. One molecule type doing both jobs.
Wow. That elegantly cuts the knot, doesn't it? The chicken and egg become one thing, RNA.
That's the beauty of it. Early life might have been based entirely on self-replicating catalytic RNA.
Is there actual evidence for this RNA world? Or is it just a neat idea?
Oh, there's strong evidence, mostly from looking at modern biology. We see molecular fossils of this ancient RNA-based system still operating today. The most stunning example is the ribosome.
The protein-making factory in our cells.
Exactly. When scientists finally mapped its structure, they made an amazing discovery. The actual catalytic core, the part that physically links amino acids together to make proteins, the peptidyltransferase center, it's made entirely of ribosomal RNA.
No protein.
The proteins are mostly on the outside, acting like scaffolding. The fundamental chemical reaction of life-making proteins is catalyzed by RNA. It's a profound echo of the RNA world.
That is amazing, like finding a steam engine part inside a modern electric car.
Kind of, yeah. And there are other natural ribozymes doing jobs in modern cells, like cutting and splicing RNA molecules. Plus, many essential coenzymes, molecules that help enzymes work, like acetyl-CoA or NADH, they either are RNA nucleotides or have RNA bits within them, suggest they originated in an RNA-based metabolism.
So remnants of RNA doing important jobs are everywhere.
They are. And finally, there's laboratory evolution. Scientists can actually create RNA molecules in the lab that act as polymerases enzymes that copy other RNA molecules.
They've made RNA that copies RNA.
Yes. Gerald Joyce's lab, among others, has evolved ribozymes that can replicate RNA with pretty good accuracy. They can even undergo Darwinian evolution in a test tube mutations happen, the better replicators take over. It shows the principle works. RNA can drive evolution on its own.
That's incredibly powerful. RNA seems like the perfect candidate molecule to kick things off, but there are always buts in science, right? What are the challenges for the RNA world?
Absolutely. It's a leading hypothesis, but it's not without significant hurdles. The biggest one is probably prebiotic synthesis and stability.
Making the RNA in the first place.
Exactly. RNA is complicated. Getting its building blocks, the nucleotides, to form under plausible early Earth conditions is tough. Especially the sugar part, ribose. Ribose is notoriously unstable in water. It falls apart easily.
So the core component is fragile.
Very.
Yeah.
And then linking the base, the sugar and the phosphate group together into a functional nucleotide without modern enzymes. That's also chemically tricky. It's often called the major unsolved problem for the RNA world. How do you get enough stable building blocks?
Okay, that's a big one. What else?
Well, there are catalytic limitations. While ribozymes can catalyze reactions, they're generally not as efficient or versatile as protein enzymes. Proteins use 20 different amino acid building blocks, giving them a much wider chemical toolkit for catalysis. RNA only has 4 bases.
So maybe not powerful enough for complex life.
Perhaps not initially, or they might have needed help. And third, there's still an information problem, just pushed back a step. Even a self-replicating ribozyme is a pretty complex specific sequence of RNA. What are the odds of that sequence arising randomly from a pool of RNA strands? It's statistically improbable.
So you solve the chicken and egg, but you still need to explain how the first functional RNA chicken egg appeared.
Exactly. This has led some people to think about a pre-RNA world, maybe using simpler, more stable genetic molecules that were easier to make, which then paved the way for RNA later.
Okay, so if RNA world has issues, especially with getting started, what's the main alternative? You mentioned metabolism first.
Right. This flips the script. Instead of starting with a replicating gene like RNA, it proposes that life began with self-sustaining networks of chemical reactions metabolism.
So chemical engine came before the blueprint.
That's the idea. Imagine, perhaps in those porous hydrothermal vents we talked about, small organic molecules getting involved in autocatalytic cycles.
Meaning the cycle makes something that helps the cycle keep going.
Precisely. Product A helps make B, B helps make C, and C helps make A again. A self-feeding loop. If such a network could harness energy from the environment, like those chemical gradients in vents, and pull in simple precursor molecules, it could essentially grow, making more of its own components.
So it would show basic life properties like using energy and maintaining itself, even without genes.
Yes, homeostasis and energy use could come first. Genetics, like RNA, might have evolved much later, maybe as a way to stabilize and reliably pass on the information inherent in these successful metabolic cycles. The doing selects for a way of remembering how to do it.
That's a fascinatingly different perspective. Genetics as an afterthought, almost.
It's a different emphasis. Now, connecting this to the bigger picture, this genetics first versus metabolism first debate, it's probably a bit of a false dichotomy.
You mean it's likely not one or the other?
Increasingly, that's what researchers think. The early Earth environment probably had building blocks for both nucleic acids and proteins, or at least peptides, short protein chains. It's unlikely one system developed in total isolation.
So maybe they grew up together.
That leads to the idea of a ribonuclear protein world, or RNP world. Co-evolution. Maybe short peptides helped stabilize early RNA molecules or acted as simple cofactors, boosting ribozyme activity. And maybe simple RNA templates started crudely directing the linking of amino acids into useful peptides.
A feedback loop. RNA helps make peptides. Peptides help RNA work better.
Exactly. A synergistic bootstrapping process. It changes the question from which came first to how did this interdependent system get started and build complexity together? It feels much more plausible, doesn't it? A messy, collaborative beginning.
It does feel more organic, less like a switch being flipped. Okay,
Yeah.
so whether it was RNA first, metabolism first, or this RNP coevolution,
Yeah.
eventually something critical had to happen next. Getting a boundary, putting wall around the chemistry.
Compartmentalization, absolutely essential. The origin of the protocell, with a membrane separating the inside from the outside. This is where the lipid revolution comes in.
Lipids.
Like fats.
Exactly. Modern cell membranes are complex, mostly phospholipids, but early membranes likely use simpler molecules, single-chain amphiphiles, like fatty acids,
Amphiphiles, meaning
meaning they have two distinct ends, a hydrophilic head that loves water and a hydrophobic tail that hates water.
like oil and water not mixing.
Precisely. And when you put these amphiphilic molecules in water, something amazing happens spontaneously. To hide their water-hating tails from the water, they self-assemble. They might form little spheres called micelles or sheets called bilayers, and those bilayers can fold around and seal up into hollow spheres called vesicles.
Tiny bubbles made of fatty acids. Essentially, yes. Primitive protocells, formed automatically by the laws of physics and chemistry. No complex machinery needed, just the molecules themselves and water.
And why was getting inside one of these bubbles such a big deal? What advantages did it offer?
Oh, huge advantages. Game-changing advantages. First, concentration. Suddenly, all your important molecules, your replicating RNA, your peptides, your building blocks, are trapped in a tiny volume, not floating randomly in an ocean. Reaction rates go way up.
Like moving from a lake to a test tube.
Perfect analogy. Second, a unique internal environment. The membrane creates a boundary, allowing the inside chemistry to be different from the outside. Maybe a different pH, different salt levels, optimized for whatever reactions needed to happen. The first step towards homeostasis, keeping things stable inside.
Controlling your own little world.
Exactly. Third, and this is absolutely crucial for evolution, coupling genotype to phenotype.
Linking the blueprint to the outcome.
Yes. Imagine an RNA molecule mutates and becomes a better replicator. If it's just floating free, that benefit might diffuse away or help its competitors. But inside a vesicle, that RNA molecule and its copies get the benefit. They replicate faster within their own compartment. The vesicle with the better RNA grows and divides faster. Natural selection can now act effectively on individual protocells.
The success stays linked to the innovator.
Precisely. And finally, these simple fatty acid membranes had selective permeability. Small nutrient molecules could seep in from the outside, while the larger important polymers like RNA and peptides were kept trapped inside. So you could get food in, but not lose your machinery.
Wow. Concentration, control, selection, resource management. Membranes seem key to everything.
They really are fundamental to cellular life.
But wait, another chicken and egg. Modern cells make their membrane lipids using enzymes that are already embedded in the membrane. It sounds like you need a membrane to make a membrane. How did the first one ever form?
That was a major puzzle. It led to this idea of membrane inheritance, that all membranes must come from pre-existing ones. But recent research has offered a stunning potential solution: de novo membrane formation.
Meaning making membranes from scratch, without a starting membrane.
Exactly. Experiments have shown that you can take simple, soluble precursor molecules like acetate, just a 2-carbon molecule plausibly abundant on early Earth, and use a minimal set of soluble enzymes, not membrane-bound ones, to build long-chain fatty acids.
Okay.
And then these fatty acids spontaneously assemble with other components into lipids, and these lipids then spontaneously self-assemble into bilayer vesicles. From simple starting materials right through to functional compartments, no pre-existing membrane needed.
So it breaks that inheritance loop. You can build the first one.
It provides an experimentally verified pathway, yes. It shows how you could get from basic metabolites to self-assembling protocells, and these de novo vesicles could encapsulate molecules, protect them.
Could they do anything else, like energy?
Remarkably, yes. Some experiments showed that if you make slightly more complex lipids this way, the resulting vesicles can actually sustain a proton gradient across their membrane. They can build up an electrical charge, like a tiny battery.
That sounds like the basis of how our cells make energy now. Chemiosmosis.
It is, the proton motive force. Finding that even these simple de novo vesicles could potentially harness energy this way is huge. It suggests that core energy mechanism might be incredibly ancient, emerging right alongside the first compartments.
Okay, we spent a lot of time in Darwin's warm little pond, the surface soup idea. But maybe that wasn't the spot at all. What about those deep-sea hydrothermal vents you mentioned earlier? The idea that life started in the dark depths.
This is a very strong contender, the hydrothermal vent hypothesis. We're talking about fissures on the ocean floor where seawater seeps down, gets heated by magma, reacts with rocks, and then vents back out, loaded with dissolved minerals and gases.
Black smokers, right?
Yeah.
Super hot, acidic stuff.
Those exist, but for origin of life theories, the focus is often more on alkaline hydrothermal vents, sometimes called white smokers, like the Lost City Field in the mid-Atlantic. These are different. They're cooler, maybe 40 to 90 degrees Celsius, warm, not boiling. And the fluids are highly alkaline, pH 9 to 11, and rich in hydrogen gas.
Okay, warm, alkaline, hydrogen-rich water coming out. Why is that promising?
Because of the gradients it creates, this warm alkaline vent fluid mixes with the surrounding Hadean seawater, which was likely cooler, slightly acidic, and rich in dissolved CO2. Where these two fluids meet, especially within the porous mineral structures the vents build, you get steep natural proton gradients.
Like a difference in acidity or electrical potential.
Exactly. A natural proton motive force, just like the one cells create across their membranes, but generated entirely by geochemistry. This gradient could have provided the energy to drive the first metabolic reactions, like reducing CO2 into simple organic molecules. The vent itself acts like a battery.
Wow, free energy built right into the geology.
And those porous mineral structures the vents form, they're full of tiny interconnected compartments. These could have acted as natural incubators, concentrating molecules and providing mineral surfaces for catalysis long before lipid membranes evolved, like inorganic protocells.
So vents offer energy, concentration, and maybe protection from surface hazards like UV and impacts.
Precisely. Continuous supply of energy and chemicals built in compartments, shielding from the harsh surface. It's a compelling package. And it doesn't depend so much on what the atmosphere was doing.
Sounds great. What are the downsides or challenges?
Well, polymerization making those long chains is still tricky in a fully watery environment, even with mineral help. The water paradox doesn't just vanish. High salt concentrations in seawater might also interfere with forming stable lipid membranes later on. And crucially, demonstrating the complete pathway from simple chemicals to polymers to encapsulated systems under realistic vent conditions in the lab is still a major challenge. We haven't quite put all the pieces together in that specific setting yet.
So promising for metabolism may be trickier for genetics and membranes.
That's a fair summary of the current state. It's an active area trying to bridge those gaps.
We've looked at Earth's surface, Earth's depths. What if the answer isn't on Earth initially? Let's talk panspermia, the idea that life or its ingredients came from space.
Right, panspermia literally seeds everywhere. A radical idea with ancient roots, actually, going back to the Greek philosopher Anaxagoras, revived scientifically by Arrhenius, and more recently championed, sometimes controversially, by Hoyle and Wickramasinghe.
So what are the different flavors of panspermia?
The main distinction is between soft and hard panspermia. Soft panspermia, or pseudopanspermia, is the idea that only the organic building blocks were delivered from space. Amino acids, nucleobases, sugars raining down on early Earth in meteorites and dust.
And we know this happened, right? The Murchison meteorite.
Absolutely. This isn't speculation anymore. We have concrete evidence that extraterrestrial delivery significantly enriched Earth's prebiotic inventory. It definitely played a role.
Okay, so that's soft panspermia. What's hard panspermia?
Lithopanspermia. This is the more sci-fi sounding idea. That fully formed living microorganisms travel between planets, probably shielded inside rocks, blasted off one planet by an impact, journeying through space and landing on another.
Like microbes hitching a ride on a meteorite from Mars to Earth.
Potentially, yes. Or even between star systems, though that's even harder.
And what makes people think this is even remotely possible?
Well, a few things. We know complex organic molecules are common in space. We know some microbes on Earth are incredibly hardy bacteria. Tardigrades that can survive vacuum, radiation, extreme temperatures, at least for a while. It shows survival in space isn't completely impossible.
And wasn't there a Martian meteorite that caused a stir?
ALH 824001. Back in 1996, scientists announced finding structures inside it that looked like fossilized nanobacteria. That claim is now highly debated. Most think the features are abiotic, but it sparked intense research and showed the idea of interplanetary life transfer was being taken seriously.
So it's not totally crazy, but what are the criticisms?
The biggest one is fundamental. It doesn't solve the origin of life. It just pushes the problem elsewhere. Okay, life started on Mars, but how did it start there? It avoids the ultimate question.
Just passes the buck.
Pretty much. And the physical challenges for lithopanspermia are enormous. Surviving the impact ejection, the long journey through space radiation, the fiery atmospheric entry, the final impact. The odds of surviving that whole sequence seem incredibly low. Possible? Maybe? Likely? Many scientists are skeptical, and it's very hard to test or falsify when it comes to the first origin event.
So extraterrestrial building blocks definitely happened. Extraterrestrial microbes seeding Earth. Much more speculative.
That's a good summary. Pseudopanspermia is established science. Lithopanspermia remains an intriguing but unproven possibility.
Okay, we have covered so much territory. Early Earth, soup, RNA, metabolism, membranes, vents, space. Let's try to pull this all together. We're tracing the path towards the first life, but ultimately, all life we know today seems to trace back to one single point, LUCA.
The last universal common ancestor, yes.
And just to be clear, LUCA wasn't the very first living thing, right?
Absolutely not. LUCA was likely the winner of an earlier evolutionary race. Many other forms of early life probably existed, but LUCA's lineage is the only one that survived and branched out to become all the bacteria, archaea, and eukarya we see today. It was already a successful, established life form.
So what can we tell about this successful ancestor looking back from today's life? What was LUCA like?
By comparing the genes and core biochemical pathways shared by all three domains of life, the universal toolkit, we can infer LUCA's characteristics. It was likely a single-celled organism, probably prokaryotic, meaning no complex internal organelles. It almost certainly used DNA as its genetic material, stored in a circular chromosome like bacteria. It had ribosomes and used the standard genetic code or something very close to it to make proteins.
So already pretty sophisticated.
Relatively speaking, yes. It had a robust metabolism, probably capable of making its own components, autotrophic, and likely lived in a hot anaerobic environment, maybe like those hydrothermal vents. It likely harnessed energy using chemiosmosis, that proton gradient mechanism. And importantly, it probably still carried remnants of the earlier RNA world, perhaps using RNA for more diverse roles than modern cells. A transitional figure, in a way.
It sounds like bits and pieces of all the theories we discussed might be reflected in LUCA.
Exactly. And that leads to a crucial point. We often hear these hypotheses, soup versus vents, RNA versus metabolism, presented as if they're competing teams and only one can win. But the reality, the most plausible scenario, is probably a hybrid model.
An integration of the different ideas.
Precisely. No single hypothesis seems to explain everything perfectly on its own. But if you weave them together, you can build a much more coherent narrative. Think of it like building a car. You need an engine, a chassis, wheels, fuel, different systems contributing to the whole.
Okay, so paint us a picture. What might this integrated story look like?
All right, let's try. You start with the raw materials, the monomers. Some are made here on Earth, maybe in localized atmospheric reactions near volcanoes or shoreline evaporation pools, a kind of modified Miller-Urey. Plus, a significant contribution arrives from space via meteorite pseudopanspermia, topping up the inventory.
Soup ingredients from multiple sources.
Yep. They accumulate in the oceans. But the key steps, concentration and polymerization, don't happen just anywhere. They happen in specific, structured, energy-rich microenvironments. Perhaps the porous, mineral-rich chimneys of alkaline hydrothermal vents?
Using the vent's energy and structure.
Right. The natural proton gradients power early metabolic cycles, fixing carbon. Mineral surfaces inside the pores concentrate monomers and catalyze their linkage into polymers, maybe symbol peptides and early informational molecules, perhaps evolving towards RNA. An RNP world starts to emerge right there, peptides and nucleic acids co-evolving.
The engine and blueprint developing together in the vent incubator.
Exactly. Then lipids, also synthesized perhaps using vent chemistry or arriving from elsewhere, self-assemble into vesicles around these active chemical systems, butting off the minnow surfaces maybe. Now you have protocells, self-contained, replicating, metabolizing units.
The first independent cells escape the vent.
Possibly. These protocells, now freed from the vent structure, can start exploring new environments, evolving further through Darwinian selection, competing, innovating, eventually leading to LUCA, the ancestor that successfully spread and diversified across the planet.
That weaves everything together quite nicely. Earth synthesis, space delivery, vent energy and structure, RNA protein coevolution, lipid encapsulation.
It acknowledges the strengths of each idea, recognizing that life's origin was likely a complex process involving multiple stages and environments, not a single magic bullet.
It's an incredible testament to scientific progress, moving from pure speculation to having these detailed, plausible, experimentally grounded models. But there's still gaps, right? Big questions remain.
Oh, absolutely. Huge gaps. We've made amazing progress, but the journey is far from complete.
So what are the major frontiers? What keeps origin of life researchers up at night?
Well, that prebiotic synthesis of nucleotides, especially ribose, remains a massive challenge for the RNA world. How did nature make enough of these crucial yet fragile building blocks? That's probably the single biggest chemical hurdle right now.
Still need to reliably make the RNA bits.
Yes. Then there's the origin of the genetic code itself. How did that specific mapping between 3-letter RNA codons and the 20 amino acids get established? It seems arbitrary, yet it's universal. Why this code and not another? It's a deep information theory problem at the heart of biology.
The translation system.
Exactly. And related to that, the emergence of homochirality. Why does life use only left-handed amino acids, L forms, and right-handed sugars, D forms? Chemistry naturally produces a 50-50 mix, racemic. How did life pick one hand and stick with it so completely? It's a fundamental symmetry breaking that we still don't fully understand.
So building blocks, the code, and the handedness are major mysteries. Where do we look for answers?
Research pushes forward on many fronts. Lab experiments are getting incredibly sophisticated, trying to evolve self-replicating RNA systems from scratch, building increasingly complex artificial protocells, testing prebiotic reaction pathways under more realistic conditions.
Trying to recreate the steps in the lab.
Yes. Plus, advances in geology and planetary science give us a better picture of early Earth conditions to guide those experiments. And then there's astrobiology.
Looking for life or signs of its origin elsewhere.
Exactly. Exploring places like Mars or the ocean moons Europa and Enceladus in the outer solar system. These moons seem to have liquid water oceans under ice shells, potentially with hydrothermal activity. Finding evidence of abiogenesis, or even just complex prebiotic chemistry happening independently somewhere else, would be revolutionary. It would tell us if life's origin is a common outcome of planetary evolution.
That would change everything.
It really would. It would give us another data point besides Earth.
What an incredible journey we've taken. From this violent primordial Earth, through chemical soups, catalytic RNAs, protocell bubbles, deep-sea vents, maybe even journeys through space. It's amazing how far science has come in piecing together this ultimate origin story. We have these plausible pathways now, backed by experiments, even if huge questions remain. It's gone from philosophy to real, tangible science.
And here's where it gets really interesting for me. Thinking about what this all means. This whole quest to understand how life started here, it's really a quest to understand the potential for life everywhere, isn't it?
Fundamentally, yes.
Yeah.
Are the conditions and processes that led to life on Earth unique, a cosmic fluke, or are they common?
Right. If abiogenesis happened here, driven by basic chemistry and physics on a wet, rocky planet with energy sources, how many other places in the universe might fit that description? What might be happening right now under the ice of Europa or Enceladus? Are those ocean worlds bubbling with their own versions of hydrothermal vents, potentially cooking up their own life?
Those are the tantalizing questions driving future exploration. It feels like unlocking the secret of our own genesis might just unlock the biggest secret of the cosmos. Are we alone? Is life a rare miracle, or is it a natural, maybe even inevitable consequence of planetary evolution? Finding out how our story began could tell us about countless other stories waiting to be discovered.
It's a truly profound motivation. And it emphasizes that this field is constantly evolving. New discoveries in chemistry, geology, astronomy, biology, they all feed back into this question. The picture is getting clearer all the time, but there's still so much to learn. So definitely keep exploring this area yourself, because the next big breakthrough could happen tomorrow, changing our understanding of life itself.